Hello, I'm Bruce Alberts, and I'm going to talk about something I spent 30 years on, trying to understand how DNA is replicated, a very fundamental process in all living systems. I'm going to give you a little history because I think that outlining the pathway to discovery where we were lost in the woods, not knowing where we were going and misunderstanding things, can help to make sense of what we now know. It also trying to convey a sense of how science works in this short talk. So this is a picture taken when I was in high school. 1953, a major breakthrough in our understanding of life happened when Watson and Crick correctly predicted the structure of DNA double helix. And as soon as they got that structure, our understanding of living things, and specifically of heredity, changed dramatically. I'd like to point out at that time, Watson was only 25 years old. I'm a big supporter of young scientists. Most creativity on the part of science happens when they're much younger than I am. This is the famous paper, first paper published on the double helix, and it has this famous sentence outlined in yellow here, that it has not escaped our notice that basically our model suggests how genetic information is transferred between one generation and the next. They were correct. Now the point is they subsequently published a short paper in the same year which basically outlined this scheme called semi-conservative DNA replication, where each old strand of the DNA double helix templates the formation of a new one using the 4 base pairs that they observed in the DNA double helix. But they had, important to note, that they had no idea how this actually would happen. They understood in principle how it could happen. And the next major breakthrough came only a few years later. A biochemist, Arthur Kornberg, who also earned the Nobel Prize, as did Watson and Crick, discovered the enzyme DNA polymerase. This enzyme was, thought to be very remarkable at the time. It will add a new nucleotide to the end of one DNA strand called the primer strand, only if that strand is paired to a complementary strand that serves as a template. And there are 2 problems. Basically, I'll describe with their finding as far as really understanding how it worked. Because they found that the template strand must be single-stranded with its bases exposed. And second of all, that the DNA polymerase enzyme moves in only one chemical direction, that is growing the 3' end of a new DNA strand. So here's a little schematic view. Primer gets added on to one nucleotide at a time. Which nucleotide gets added depends on what base is on the template strand at that point. And the 3' end of the primer extends one nucleotide at a time. So this is a more detailed view showing some of the chemistry. We actually, as discovered by Kornberg, the source for the energy of polymerization is a incoming deoxyribonucleoside triphosphate, and 2 phosphates marked in red get split off every time this single unit is added to the 3' end. And again, as you better see here, which base is added depends on specific base pairing. Here, C with a G. The G is on the template, C is coming in as a new monomer. So I started working on DNA replication way back then. I was a beginning graduate student, and I was fascinated by trying to solve the problem of how DNA might replicate. What was the machinery that would carry out the process that Watson and Crick predicted? And one of my professors, a famous professor, talked about, I still remember him giving us a lecture about how it might work. And here's one of the models that was suggested. Since the DNA polymerase only goes in one direction, maybe replication only starts at the one end of a chromosome, and opposite ends get synthesized going along in this mechanism diagrammed here. And I just want to show you this would create a lot of single-stranded DNA, because this strand opposite this template strand would be released into a DNA single strand. And so if you looked at a replicating chromosome, you should see a lot of single-stranded DNA. Well, just 1 year later, a man by the name of John Cairns did a famous experiment where he actually did electron microscopy of what you see when DNA is replicated, and he discovered the replication fork. That is, there isn't any of this large amounts of single-stranded DNA. Instead, DNA is replicated on both sides, both strands of DNA double helix roughly at the same time. And so at a replication fork, the two daughter DNA strands appear to grow at opposite ends, and yet we know that DNA polymerase only goes one way. How is this possible? This was the mystery that faced me when I started to work in the lab as a graduate student. And this was the way people drew it, actually. Same professor is famous for drawing a fig leaf a little later over the replication fork, saying we had no idea what happened at that point. So in 1961, when I started my work as a graduate student, and when I did this, I was given the freedom to choose anything I wanted to work on. That was wonderful. And I wanted to solve the mystery of how DNA replicated. We assumed, everybody assumed, that a single enzyme, DNA polymerase, alone replicated DNA. But how was this possible? Two problems, of course, that DNA polymerase works only on single-stranded DNA, and yet DNA is a double helix inside the cell, as Watson and Crick showed. And secondly, the two DNA strands, again, as Watson and Crick showed, run in opposite directions. They're anti-parallel. And yet the polymerase only goes one way. So this led to a very unsuccessful career as a graduate student, because I had been inspired by Watson and Crick to believe that theoretical biology could predict things. And I think not only me, but many graduate students were predicting things and trying to test their ideas. We're more sophisticated now. So I predicted how DNA polymerase might be able to start DNA synthesis on a chromosome. I actually used it as a term paper in one of my graduate courses, and I got an A+. So, oh my goodness, this must be right. So I then spent years doing experiments trying to see if my theory was right. Of course, my theory wasn't right. And as you'll see, the whole hypothesis was wrong that one enzyme can replicate DNA. And I had a very unsuccessful, difficult time as a graduate student, from which I learned a lot. You could hear about that if you go to the short talk I've given on iBio as well. It's also here in the section about lessons learned from failure. And I've had many failures. That's only one of them. I explained how failure is important for a career. You have to learn from your failures. That's what makes you better the next time. But then I finally got my PhD, and I moved to Geneva, Switzerland for a year as a postdoctoral fellow, and I immediately discovered, to my great surprise and somewhat consternation, that had I been reading the scientific literature, I would have known better, because work done in bacteriophage T4, a wonderful model system for early stages of molecular biology. It's a large double-stranded chromosome. The virus infects E. coli, and it makes all its own replication machinery. And it had about 100 genes discovered by genetics and published in 1963 in the middle of my thesis. And the fact is that there were at least 7 genes of this virus that were absolutely required for DNA replication of this virus. The geneticists who did this work had no idea what these genes did, and they gave them numbers. 32, 41, 43, 44, 45, 61, and 62. A little later, one of these genes, the gene 43 protein, had been shown to produce the T4 bacteriophage DNA polymerase, so that we understood, that made sense. But the question was, why are these 6 additional proteins needed for any replication of the T4 chromosome when the virus infects E. coli? And this meant that I and many others had been completely naive in thinking that only a single protein was needed. These 7 proteins must be needed, and therefore replication must be much more complicated than anyone had imagined. So then I was getting ready to start my old lab, and I needed a strategy. And how was I going to, every starting scientist really should have the opportunity to start a new problem from scratch. That's the way we get innovation. And again, I was still devoted to solving this problem that I had failed to solve, and now I had a new strategy, a completely different one. I decided I'd make a column out of DNA and looked at all the proteins that bind to DNA that in that column. As a biochemist, you break open a cell and pass the crude extract over the column, and some of the proteins, a small fraction of them, will bind to the DNA column. They could be then removed from the DNA column by raising the salt concentration. And I found that many proteins in a T4 infected cell made by the T4 bacteriophage genes bound to this column. Then the question was, would one of these proteins be missing if a mutant virus is used instead of the normal virus for the infection? If so, then I could find a protein that the genetics said was needed for the then very mysterious machinery that replicated DNA. Here's a slide taken from an experiment we published in the Cold Spring Harbor Symposium in 1968, the first real discovery from my independent lab at that time at Princeton. We had done that experiment I just described and found that when you infected a cell with a gene 32 mutant, a major band that was present in all the other infections was missing. Completely obvious. And because it was missing only when you had a gene 32 mutant, we concluded that that must be the product of gene 32, called a 32 protein, and that therefore it must also be required for DNA synthesis. But of course, we had no idea what it did, so we had to do biochemistry. And basically, what we discovered is drawn in this little schematic. The 32 protein binds tightly and cooperatively to single-strand DNA, forming long clusters on the DNA. In so doing, it straightens out the DNA molecule because DNA, single-stranded DNA, like single-stranded RNA, would otherwise be folded into little knots because these little hairpin helices that can form with imperfect base pair. And this open form of DNA is the kind of DNA that DNA polymerase could more effectively use as a template. So here's one of our great experiments. Seeing is believing. I collaborated with electron microscopist Hayo Delius at Cold Spring Harbor, and here's what we found. When we took circular DNA single strands, this is actually the viral DNA from a small virus, but it's completely circular and single-stranded. And without 32 protein, it's this little ball. You see an example with that arrow. Without 32 protein, it's a little ball all tangled up in a little knot. When you add 32 protein, the whole molecule is extended in a beautiful way, in a form that turns out leaves the DNA bases exposed so that DNA polymerase could still see them. So if we now go to the next slide, this is what we conclude after doing a number of experiments, that the form in which the 32 protein holds the DNA is a much better template for DNA polymerase, allowing it to go much more rapidly than it would go otherwise over this structured single-stranded DNA, which would be the form of DNA inside the cell without the 32 protein. Now there were 5 mysterious proteins left. Of course, we looked for some of the other mutant bands on our columns. We didn't see them. So we tried a different approach called an in vitro complementation assay. Here's a very simple idea. If you break open an infected cell and add precursors to DNA, you see that some of that precursor nucleotides are incorporated into DNA, a very little amount. It's a very bad reaction because everything's being degraded as it's being made and everything's mixed together inappropriately. But with that assay, we asked, could we find a stimulation of DNA synthesis in this crude mixture that depended on adding one of the 5 missing proteins? And fortunately for us, it worked for all the missing proteins. And this is the kind of data we got. Basically, if you added any extract of an infected cell to a crude lysate made with a mutant, say in this case it was 62 mutant, you could get a little stimulation very rapidly stopped with this lousy DNA synthesis. Twofold stimulation amount of DNA made. If you added the original extract, of course, or uninfected cell, nothing happened. So we had an assay. And as soon as a biochemist has an assay, you could purify anything by following it through a series of chromatography steps. This work was mostly done by my graduate students at Princeton. And finally, after years of collecting each of these proteins, every time we got a new one, we added to DNA polymerase and see if it would replicate. The answer was no, but when we had all 7, and only when we had mixed all 7, suddenly we had this magic day. Which, remember, I've been working on this since 1960, very unsuccessfully. Now we could do what I could never do before, get replicated double helical DNA template. If we left out any one of the 7 proteins, it didn't work. Now, the important point here is that this kind of in vitro biochemistry with purified proteins is essential if we're going to understand how life works, because only then can you manipulate the system to actually see what actually is going on. And it took us another 6 years then to figure out why each of the proteins was needed in this reaction. I'm not going to go through all the details by any means. I'll just give you one example that's simple. The gene 45 protein turned out to form a sliding clamp for the DNA polymerase, which allowed the polymerase to move for many thousands of nucleotides, hundreds of thousands, without dissociating. This is how we viewed DNA replication in the old days. This is how we viewed life in the old days. Basically, everything was a random collision. And so here's a DNA polymerase coming in, adding one nucleotide, falling off, a second DNA polymerase, because collisions are very rapid inside of a cell in chemistry. We knew from chemistry. So the second DNA polymerase comes in, it adds another one, then it falls off, another one comes in. Well, it turns out that kind of mechanism for life in general is so inefficient relative to what actually happens that it's only rarely used. Usually things are much more organized, as I'll explain. It turned out that the 45 protein produced a sliding clamp for the DNA polymerase that we could show biochemically enabled the DNA polymerase to go for hundreds of thousands of nucleotides. We tried to figure out why that was so, and I had a graduate student who worked very hard on this. But we missed the fact that it's first discovered in the E. coli system by Mike O'Donnell. The reason why it worked like this is it was a ring formed around the DNA that created a tether for the DNA polymerase. And the DNA polymerase is kept associated with the same DNA molecule because it's pulling this ring along as it moves. And this is a structure that was solved originally for the E. coli protein. I pointed out on several slides that there are human versions of all these proteins. In this case, that human version is called pCNA. It looks the same, a ring pulled along by DNA polymerase. So I skipped the important point here because I was talking about what I was doing. But early in our work, by 1970 or so, we knew from the work of a lab in Japan, 2 Okazaki, husband and wife, and their co-workers, that when you looked at the newly synthesized DNA inside of a cell, Actually, they used radioactive pulse labeling to see what the new DNA was like. The DNA, half the DNA, that made on the so-called lagging strand, the strand that was so mysterious how you could move at a replication fork. That DNA is made backwards. Again, it's made by the same DNA polymerase, which can now go backwards because that's the direction it goes. But it's made as a short series of fragments which are subsequently run into each other and sealed together to create a continuous strand on the lagging strand of the DNA replication fork, just like the DNA on the leading strand is continuous because it's made continuous. Now, this is a mechanism that was completely unpredicted. I think if anybody had predicted this mechanism in 1960, they would have said you're crazy because this was too sloppy. It would create a mess. All these fragments would get mixed up and you'd have all kinds of errors. But in fact, this is the way life works. More specifically, each Okazaki fragment, although it's made by the same DNA polymerase on the leading strand, that DNA polymerase needs a special primer to get started. And that primer must be made very frequently on the lagging strand to start each of the Okazaki fragments, every 100 or 1,000 nucleotides, depending on the system. And this requires a separate enzyme. It turned out to be one of the 7 proteins that was so mysterious in our system. This enzyme is called DNA primase. It makes a short fragment. The DNA polymerase then takes over. Very importantly, because you don't want RNA in your final DNA product, that RNA is recognized and specifically erased before all these fragments are sealed together in the end to make a continuous leading strand. Now, in retrospect, we could understand why DNA replication is so complicated. We never predicted it. It's because of the need for enormous accuracy. DNA polymerase turns out to have 2 active sites, one for polymerization and one for proofreading. Why is proofreading needed? Proofreading is needed because although we might imagine that the base pairing that Watson and Crick discovered would be perfect, A could only pair with D and C could only pair with G. This violates all the rules of chemistry, and life must exist with chemistry as it is in the real world. And it turns out that you can't have perfection in chemistry. And bases have rare tautomeric forms, which are of higher energy, but nevertheless exist at 1 part in 10 to the 4th or so, 1 part in 10,000. And in their incorrect minor tautomeric form, they could pair incorrectly. And for example, a C could pair with an A and be added by DNA polymerase as it's moving. DNA polymerase evolved so it could recognize that mistake, because before it will add on to the previous place where it just added, it makes sure that that 3' end is base paired. And so, as indicated in this diagram, it can recognize that something's wrong, back up, chop off the mistake it made, and go forward again. And this turns out to increase the accuracy by something like a factor of 100 over what would happen without a self-correcting polymerase. Anyway, to make a long story short, I'm not explaining this really, only the enzyme that goes in this direction can do that effectively. We understand that, and that's why we don't have a DNA polymerase going continuously on the lagging strand. And it's also why DNA polymerase must have a primer to start. It can't start a new chain by itself because it looks for a perfect pairing before it can start, and thus also explains why an erasable RNA primer makes so much sense and why you need a DNA primase enzyme. Well, the net result of all this, plus things I can't talk about, is DNA is replicated with only about 1 mistake per billion base pairs that are copied, 1 part in 10 to the 9th. This is incredible. Humans can't do anything like this. And we know that without such accuracy, a complex organism like a human could not exist. We'd have too many mutations. One of the things that would happen is every embryo would get cancer and be destroyed. Many other things would go wrong. And so what we could imagine is that as cells evolved to more and more complex forms, all these mechanisms got added. And somehow the original mechanism by which DNA replicated obviously couldn't have been this complicated. But after all these many, many rounds of evolution, billions of years of natural selection, we ended up with the mechanism that I was fortunate to be involved in helping to discover, which was, of course, a real privilege and a great pleasure to solve this mystery. So here's the DNA replication fork with its proteins on it. I didn't even talk about some of these. I talked about most of them. The two green molecules are the DNA polymerase, one on the leading strand and one on the lagging strand. Same in our system, it's exactly the same DNA polymerase molecule. The red ring behind it is the sliding clamp that keeps the polymerase from dissociating from the DNA. The DNA polymerase is tethered by this clamp to the DNA. On the leading strand, this is all very simple. It just keeps going. On the lagging strand, it's very complex. It stops and starts every few hundred nucleotides in human cells. And every time you want to start a new one, you have to make a new RNA primer. And that involves an enzyme, which is one of our 7 proteins, called a DNA primase, special protein that makes a short RNA primer. A few nucleotides long, just long enough to start the synthesis by the DNA polymerase each time. You can see the single-strand DNA binding protein coating all the single-stranded DNA. It's probably also ahead of the DNA polymerase molecule on the lagging strand, not indicated there. The new molecule I haven't described is, again, one of our 7 proteins was an enzyme called a DNA helicase. Very important enzyme which uses ATP energy, hydrolyzing ATP to propel itself along the DNA and opening up the helix ahead of the leading strand DNA polymerase to make the DNA single-stranded in the form that the DNA polymerase needs in order to copy that template. Now, the fork is actually folded. This was probably my most exciting discovery, realization. Basically, I was giving a seminar somewhere, and I was, like I'm doing now, and I suddenly realized that it made no sense for the DNA polymerase on the lagging strand to have to come on and off every time it made an Okazaki fragment, that I could actually test whether, as a biochemist, whether it was being retained and used over and over again, and we actually found that was true. So the fork is actually folded to allow the DNA polymerase on the lagging strand to be used over and over again, therefore allowing very rapid DNA synthesis. So really, the whole point of my talk really is to introduce you to this movie, my favorite movie. It was not made by me. It was made at Cold Spring Harbor Laboratory by Bruce Stillman and co-workers who work on human and yeast DNA replication. It was part of the 50-year DNA celebration there, 2003. There was a big party. I was there. And this movie was produced and distributed for everybody to use, originally through the Cold Spring Harbor website. And I'll let this movie play. It's narrated as it's narrated in our textbook. And you can find this movie on YouTube if you want, as well as on my talk. So this shows the replication fork in action. And what I want you to get from this movie is the general idea about how all of life works. It's life. I could talk to you only because life, the biochemistry of life, and the chemistry of life is this complicated and elegant.
Using computer animation based on molecular research, we are able to picture how DNA is replicated in living cells. You are looking at an assembly line of miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called a helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
Well, that movie I find inspiring. It really shows this mechanism that we have been working on for so many years, me and many other laboratories. And it's so satisfying to work on mysteries for decades and finally solve mysteries. So that's why it's my favorite movie. So it now turns out that almost every process in a cell is recognized to be driven in a similar way with different mechanism by a complex of 10 or more proteins that works like what we call a protein machine. These protein machines function very much like the machines in everyday life that are driven by electric energy, except that, of course, they're not driven that way. They also need ordered movements, in this case, ordered movements of proteins and RNA molecules, and those ordered movements are driven by proteins in the set that specifically hydrolyze a nucleotide, ATP or GTP, to give it unidirectional motion and harness the energy of that hydrolysis to make the machine be effective. Because you need ordered movements to make machines. And that's why we use electricity to drive machines in everyday life. So here's some, now at the end of my talk, this is basically some personal lessons that I learned as a scientist. I guess the most important one is not listed here, which is that being a scientist is exciting. You get paid for solving problems that are both fascinating to try to solve, puzzles, but also in the end turn out to be useful because although we weren't intending this, the kind of information we discovered is very important for understanding of what happens in human diseases where these things go wrong, including cancer. But the major mystery, the major point is that to work on something that it's important that you don't understand. I call them mysteries, because if you then solve that problem, it will help the whole world understand whatever process your process is connected with much better. I mean, obviously we needed to know how DNA replicated. That we didn't know what it'd be useful for, but it obviously has helped us understand many other things. Second thing, which is I've been stressing, and it often gets lost in this push to go take government grant money and solve some disease problem, is that the shortest path to actually solving those kinds of human problems often lies in backing up a bit and coming to understand how basic processes work. And this is often best done not in human cells, but in a model organism like the one I use, or a bacteria, or a yeast cell, all of which are much more readily manipulable and easier to understand than our human cells. And finally, of course, the major lesson from my PhD thesis is that in many ways the Watson and Crick discovery, which was so critical, was unique. Most theoretical predictions of biologists turn out not to be correct and to be oversimplified, because nearly all cell processes will be based on elegant mechanisms which are too hard to predict. You just have to go and find out how that works by experiment. So thank you for listening. I hope this helped you to better understand a basic process that's going on as every minute of the day in billions of the cells in your body. If it wasn't going on, of course, you'd be in very bad trouble. You couldn't be alive. Thank you.